We are going to show you today um, some of the experiments of the Biophotonics box. So even for some of you who might not receive the kit if you are not part of the internship program or the CPD program we are offering at IPIC, you can do most of the experiments even uh, even with very cheap components that we have with the proposed experiments. So can everyone actually see my screen? Okay,
Yes.
that's perfect. So yes, so today we will discuss about the experiments then that we have proposed so that you can explore biophotonics at home. So we have the manuals of these experiments available in the QR codes. You can access them through the QR codes of our workshop agenda. You can just scan them with your smartphone and it will redirect you to the link of where we have our manuals. Now we have our manuals from the last year and we will aim to update this by next week so that when we expect the Biophotonics kits to have arrived to everyone participating in the programs offered by IPIC. And here I also put, you can find them in the agenda there. And here I also make available the links that you can use to access both the manuals of the Biophotonics box experiments and the Biophotonics computer simulations we'll also be discussing in this workshop. And today would like to discuss with just the experiments we have. So the LED circuit experiments with light penetration and fluorescence, the experiments with tissue mimicking phantoms, so you can actually mimic the tissue with the optical properties it has, and also look at very small things with your smartphone, so you can make a microscope out of it. And when we are going through all of that, we'd like you to keep in mind that how is the relationship between the experiments with the concepts we saw yesterday and what you see in the webinars. We have three key components of the workshop, which will be the theory on the webinars, will be the experiments you can do at home, and the computer simulations you'll be doing in one of the sessions. And these will be the integral part. We make them as related as possible, and we would like you to explore the benefits of each of them for your own interests and looking at different backgrounds, as you saw that Stefan was saying, that we need people with several and very diverse backgrounds to do biophotonics. So we will start by looking at the LED circuit experiments with Kasia. So I hand over to you, Kasia, and you let me know when I need to pass these slides.
Thank you, Marcelo. Hi all, so my name is Kasia and I'll just briefly walk you through some experiments and activities that we planned for you with the LED circuit board. So when you receive your Biophotonics kit, you will find a lot of components inside, like for example, solderless circuit board, which you will use to build a circuit that will power different colors of LEDs. So you will find also some resistors there and cables and the batteries and different colors LEDs and a step-by-step instruction on how to put it together in order to make it work. So this component will be part of many of the experiments within the Biophotonics kit. So I hope that the instructions will be clear how you should put it all together, okay? Next slide, Marcelo, please. So the first activity that we planned for you is exploration of penetration depth of different wavelengths. So you can see on the left-hand side of the screen a picture of one of the people from our group holding her finger on top of the 3 different color LEDs. And you can see that all the colors propagate a little bit differently. So we would like you to explore that in a bit more detail. And what you might find useful is the absorption spectra from different chromophores that you can encounter in a biological tissue, like for example, hemoglobin, water, or lipids. that you can see on the graph on the right-hand side of the slide. So what you might think about is why, for example, the red color is propagating deeper. What are the reasons behind different wavelengths propagating different distance? And what other wavelengths would be the most suitable for deep tissue penetration? Next slide, please, Marcelo. So the next phenomena that you will explore is fluorescence. So on the right-hand side of the slide, you can see the Jablonski diagram, which shows how the fluorescence process works. So we have an absorption of a high energetic photons, and then part of that energy will be re-emitted as in the form of fluorescence by a lower energy photon. So what we will try to explore here is using the LEDs that we provided to you with the board, and see what kind of everyday objects you can find emitting light. So we will start with bananas, for example, and you might want to try to figure out why bananas might fluoresce under UV illumination. We will also look at laundry detergent or tonic water and any other really household items that you can find that emit light. And also, for example, how the sunscreen actually works. So all of these experiments will be described in a bit more detail in the manuals that you're going to get that Marcelo mentioned before. But this is also kind of up to you. You can explore, I think, many more different things using the LED circuit board there. Next slide, Marcelo, please. So the last experiment that I will talk about explores light propagation and light scattering. So you will have to download the Firefox app, which is free of charge. And that app allows you to explore different physical properties just using your phone sensors. So the light sensor that we will explore in this experiment is typically placed on the front of your smartphone, just next to your front camera, but that might be model dependent, so you have to figure out where it is in the case of your phone. And then we will use the light function of that app to record the changes in the light intensity. So what you will need for this experiment is only water and some milk, and maybe a dark place to be able to observe the effects a little bit better. So what we will do is look at changes in the light intensity while passing through different solutions of water and milk, and try to quantify that and see how adding more milk into water will change the amount of light that we can see on the other side of the box. So maybe the last thing that I would like to mention here is also that the LEDs, they are a little bit low power. So for some of the experiments, you might need to really find a darker place to be able to observe the effects of fluorescence, for example, or the light propagation. And also that because they are low power, they are kind of eye safe, but please do not shine them directly into your eyes. So that's all from me. Back to you, Marcelo.
That's perfect, Kasia. Thank you so much for the very good explanation on the LED circuit experiments. So actually, even using the Firefox app, you can look at the light intensity at very different configurations. You can look at reflectance measurements, transmitted light, and so on. So please really explore that. And with the tissue mimicking phantom experiments, we actually start the manual by giving you a teaser with try to think how transparent a glue stick is. Have you seen how the light propagates through a glue stick? It looks reasonably transparent, but could you see how much light will end up on the other side of it? Could you actually explain why you would see different colors when you see it from the sides if you illuminate it with just a white sides, if you illuminate it with just a white light from the flashlight in your smartphone? So try to think why this actually happens. And to go through these things, we also want you to make some of the phantoms, which are basically media, which you can make with mixtures of absorbers and scatterers. In our case, we just use food coloring and milk. So very simple mixtures. You can just use them with, mix them in a clear shot glass, and you use just a very little amount of samples. So you can observe how is actually the light propagation once you have only absorption or only scattering. Like, and obviously the combination of these two, which will mimic your tissue, which has these two optical properties here. And you can also think, how exactly could I mimic tissue? Tissue have a combination of absorption and scattering properties there. Could I actually explain, predict how much light will propagate through media with more or less quantity of absorption and scattering? Could I actually predict how colors will be distributed once the light propagates within the phantoms I have made? Could actually I mix them in a way that I have a similar amount of light intensity reflected from the medium, from the phantom surface, and my hand, for example, or any other tissues we find in our body? How exactly could I predict the behavior of light propagation in these phantoms? So you could not really take all the blood from your finger in order to measure just the scattering of it, or you just can't get rid of just all your organelles or your cells of your finger if you want to measure just the absorption. But you can simulate the tissue optical properties by doing a simple experiment just by mixing food coloring and milk. So think about all these things in the back of your head, and you can explore so much here. Now, we also propose that you look at illumination at several angles or distances, and it may be a bit awkward to make it with just liquid phantoms, because it can spill out, it can make a mess in your kitchen or whatever you are doing the experiment. But we have a solution for you. We actually can make them solid. So we provide gelatin to you so you can make them solid for a few days. And you can actually illuminate it from all the configurations you want to explore the light propagation within the medium you want. So one other thing that you can do is you can insert an object with different optical properties compared to your phantom. And this object will simulate your tumor. It could be a candy, it could be a dark bead, or something that is different from the surrounding medium. And you can do a similar experiment as Sanatana was showing to you yesterday. You can see if you can find the tumor inside your phantom. You can see if the shadows of it, you can see the shadows of just a medium with just absorption or just scattering or a mix of two. How much milk, how much food coloring do you need to add in order to not see the tumor anymore? From how much scattering and absorption would you be unable to localize the tumor? And you can do this with just purely environmental ambient light. You can see if you can find the tumor by actually illuminating it with your smartphone flashlight, or even use the LED circuit we have given you to illuminate it with different colors. Could you actually find the location of the tumor better if you illuminate with just red or green or blue light? What would this look like? Could you do tomography with these? So if you are a doctor, what is the safety margin? How much tissue would needs to be around your tumor in order for you to completely remove it? If you were the doctor, how much distance from the tumor would you take to remove that tumor? So this can be a difficult decision if you are doing brain surgery, for example. You can't remove too much normal tissue from the sides. it can be very dangerous for the patient. The brain is a very sensitive part of our body. So think about all these things when you are doing the experiments. And finally, we also want you to explore how is the light propagation with different colors of light. So use your LED circuits to see what's the light propagation with, is just scattering or just absorption. You'll be doing spectral measurements, as we were discussing yesterday. You will be looking at mostly the tissue optical properties at blue, green, and red light, at red wavelengths. So you can look at how much this will influence the light propagation in your phantoms. So keep that in mind. This will relate to many of the concepts you see during our workshop. And now I would like to hand over to Baptiste, who will go over the smartphone microscope experiments for us to see very small things.
Hello, thank you, Marcelo. So can you go back to the first slide? Okay, so we'll just explore, and this is a simple activity that actually requires probably the least equipment of all, because you just need a toothpick or something small and sharp to explore, like, what can you do with a droplet of water and use the properties of a droplet of water to actually make a magnifying glass for your phone and revealing smaller things that your phone is usually, the phone on your camera is usually able to see. So just a small procedure here. If you do that picture, is if you wet a bit your toothpick and then you try to deposit, you can use it to deposit a small drop of water on top of the camera. So here I use the front camera of my phone, and eventually the sensor, so which has its normal lens in front of it, will be changed with a sensor with the normal lens and a drop of water. And since it has that lens shape and water has different refractive index than air, is going to bend the light and make it change the normal magnification of your phone. So if you can go to the next slide. So what's happening is, if you imagine the effect of the water droplet is in normal functioning, you basically have the real object that is imaged on the sensor. But now when you put the droplet of water, the real object first goes through the water, it creates a virtual object that is magnified. And then that's that virtual object that is it's an equivalent vision of what's happening. It's like you have the virtual object, it's just further, like at the same distance of the real object, but just bigger. And that's how this is happening. And that's enables you to make some sort of the first some sort of microscope on your phone, which, when you think of it, probably the microscope was the first optical tool that I use for life science and biological applications. It's the very, very old technology. And so, yeah, can go to the next slide. I tried to take a few pictures using my phone, so you can see you can go to the next slide, Marcelo. So the first one would be like a fingerprint on my hand. The second one, it'd be a leaf, so that you can start seeing in more detail the canals in a leaf. So it was a basil leaf. Then, like you see, you can start to see the pores of your skin. And then on the fourth image is just a piece of paper, so you can start seeing a bit like the different structures that are scattering the light. But feel free to observe anything you find around you. So for example, if you go on the next slide, I'll just leave you with a small question. That's a video of an animation, but just don't play it now, Marcelo. Now I'll play it when we meet again after the quiz. It's like, what can you see on a banknote? So you can try to look in a euro banknote, and there is some writings written as a safety measures on the banknote, and you can try to start seeing them by using that microscope. So yeah, so that's it for that main activity. it might require a few tries to actually put the water, a good droplet of water on the objective of your phone. But once you get the hang of it, then it's quite fun. It works quite well. And then if you want, you can also try with other liquids to see if it has an influence, like honey or oil, depending on how dirty you want to be. Oil can be a bit messy, so yeah, that's it.
That's perfect, Baptiste. I think now we have Jackie just explaining the relationship between the experiments and the computer simulations and how they can be useful in biophotonics. So handing over to you, Jackie.
Hi, everyone. My name is Dr. Jacqueline Gunther, but I usually go by Jackie. I don't know if, wait a second. Is this working? Yeah, it's working. So we're gonna meet again during, I think, the 6th webinar or the 6th session of this. And we're gonna go over computer simulations. So the question is, can we simulate light measurements? So usually how that all starts is you have some sort of system here. And in this particular system, we're actually separating the light into different wavelengths. So this is kind of similar to what you're doing with the different LEDs. You have 3 LEDs, possibly 4, that have different wavelengths of light. And that light, what we do is we put that through a sample down here, and eventually that all gets detected. So depending on the wavelength of light, you're going to have, the light's going to interact with the substance differently. So for example, here on the right-hand side, I have this graph where we have wavelengths as our x-axis and something called absorption coefficient as our y-axis. And what you can see here is oxy and deoxyhemoglobin spectra. And what we notice here is that the 2 different, what we call chromophores, actually react differently depending on what wavelength. So this is one important piece of information to know moving forward in order to simulate light measurements, because the first thing that we need to know is how the light interacts with the sample, and this is actually dependent on wavelength. Marcelo, can you go to the next slide, please? So there's 2 very important aspects of light interaction with tissue that you need to understand in order for us to be able to model this very well. So we have something called scattering. So how light bounces through the materials. This is essentially light hits a particle and bounces in a different direction. So this is called scattering, and we can actually measure this and give it a metric. And this is usually caused by the structure within your cells, or just the microstructure of the tissue. So things like cell membranes, mitochondria, the nuclei of the cell will all cause the light to bounce and scatter around. The other important aspect to know, which I just briefly mentioned before, is absorption. So how much the light attenuates as it moves through a material. So the light can start out, really bright, shine through a material, and it will come out very weak. So this is caused by different biomolecules that will actually absorb the light. And so our major biomolecules that we'll be dealing with are oxy and deoxyhemoglobin, lipid, and water. There's other ones such as bile, bilirubin, and melanin, and things like that. But the 4 major ones are usually oxy, deoxyhemoglobin, lipid, and water. And so I showed you part of the spectrum before with oxy and deoxyhemoglobin, but each of these different components, so lipid and water even, have different spectra or different ways they interact with the light depending on the wavelength. So if we look at oxy and deoxyhemoglobin over here, they have high absorption in, say, 400 to 600 range, but lower absorption at the higher wavelengths. And then lipid and water have lower absorption at the more visible region, so between 400 and say 800, but then they begin to increase as wavelength increases. So the wavelength actually plays a very important role when it comes to not only understanding biomedical optics, but when it comes to computer simulations as well. Marcelo, can you move on to the next slide, please? So why are computer simulations important? Well, it helps you do a couple of things. First, you can run multiple simulations instead of running multiple measurements. It can also help you with system design, and it allows you to model things that are difficult to measure. So, well, I'll get into that in a moment. Marcelo, can you, thank you. So I had shown you this graph before. So we have our oxy and our deoxyhemoglobin absorption. So we have this wavelength versus our coefficient. And say I want to know what the spectrum of tissue is going to look like at a particular oxygen saturation. So oxygen saturation is denoted by SO subscript 2. So I need a model, and my model is going to be something simple, such as this equation here. So I know what mu A oxy is based on the graph on the left, and I know what mu A deoxy is based on the graph on the left. And now I want to see what the mu A total is depending on the oxygen saturation. So my choices are get a bunch of vials and make sure they have different oxygen saturations, measure them, and then calculate the spectra. Or I could just use this equation when I already know this information to the left, and I can figure out the oxygen saturation absorption. So I've plotted many here, but essentially it goes from 0% oxygen, so this top line, to 100% oxygen, which is this bottom line, and then everything in between in groups of 5. So this could be run fairly quickly on a computer instead of being in the lab all day trying to get the measurements. So that's one way that computer simulations can help us. Then, yes, thank you, Marcel. Another thing that computer simulations can help us with is system design. So this is a system we designed in the lab. It's a little cuvette holder to hold a sample, and it will tell us our optical properties. But before we were able to create this system, we needed to know something very important. We needed to know the source detector distance of the fibers. So they're fairly close together. They're between, I don't know, 50 to 600 micrometers apart. But we needed to know which source detector separation would give us the best results. So one of the ways we could have done that is by a fiber that had each different set of source detector distances, which could get quite expensive, or you can run a computer simulation, just such as I did over here. So this computer simulation, what it does is it actually models how light travels through not only tissue, but different mediums. So we were working with phantoms, which you'll be working with as well, just combining milk and water. And so what I would do is I would change the different source detector distances from here to here and see how much of a, like, say, a depth I would get and how well my output signals would be over here. So I would do that for multiple source detector distances and then see which of the results gave me the best, which source detector distance gave me the best result. And that's the fiber we decided to use for the system. Next slide, Marcelo, please. Oh, the detector got moved. The detector is supposed to be up here. Not sure what happened. Anyway, so you're supposed to have a source here and then the detector over here, and this is your source detector distance. But anyway, another thing that you could do is model things that are difficult to measure. So in this particular paper, we were modeling how much light would get to a fetus that was still in your utero. So we have this is just a sample of the model, so how the model is structured. So we have a couple of layers that represent the mother, and then a couple of layers down here that represent the fetus. Now you can imagine that this is quite difficult to measure in real life. Not only that, but what we wanted to do was change how deep the fetus was within the mother and see what sort of result you would get on the detector, which belongs up here, not down there. So what we would get here is our results. So source detector distance versus the signal on the detector. We would run a light propagation model. So essentially the output is the signal on the detector. And we compared this to different fetal depths, so how deep it is from the surface of the mother. The other thing that we did was we looked at the amniotic fluid layer, which is a very interesting layer because unlike some of the other stuff that you'll be doing, it's like water. I can't draw here, but that's okay. It's right here. It's very, very clear. So it's not going to have as much scattering as biological tissue. So what we wanted to do is if we remove that layer, what sort of results we would get. And now the reason why we wanted to do this is because if we were to say monitor the fetus using this particular type of technology before labor, there would be a bunch of amniotic fluid. But once labor began, a lot of that amniotic fluid would disappear. So if we wanted to see how that would affect the results over here. So the dashed lines are with amniotic fluid and the solid lines are without amniotic fluid. And we can see that there's more signal actually when there's this amniotic fluid layer. Now, you can't actually, I guess you could, measure this in the clinic. You would need to have enough women that showed you different fetal depths, and you would have to measure them before and after, or before and during labor. But that's a lot more difficult to do than just sitting at your computer and running a bunch of computer simulations. So this is just some of the examples of why computer simulations can be super useful to us. And I'll show you how to run some of them at a future session. So that's it. Thanks, Marcelo.
Thank you, Jackie, for the brilliant presentation. So any questions, just feel free to contact us and we reach out to you. So, and thank you very much for coming. And very appreciated. I hope you enjoyed.